Thank you, Chana. Ladies and gentlemen, good afternoon. It's a great pleasure for me today to present some of ongoing work on biomarkers, biochemical and microparticle in QCRC. I will start my presentation by general definition of biomarkers, their the characteristic of an ideal biomarkers, and I will talk on microparticle specifically, the relation to pathophysiology and heart failure, and I will give an overview of the ongoing biomarkers project in QCRC and future directions. In 1989, the NIH defined a biomarker as a characteristic that is objectively measured as indicator of normal biological processes, pathogenic processes, or pharmacologic responses to therapeutic intervention. The importance of biomarkers is reflected by the increased number of publication and studies made on biomarkers over the last 20 years. And in the last 5 years, we have witnessed a huge number of publications which reach an average of 700 publications per year. So what are the characteristics of an ideal biomarker? So before we can use it in clinic, there is some characteristic like a biomarker should be available and easy to measure, and it should have a defined release kinetics and designed to answer a specific question in heart failure and to give information about the prediction, the diagnosis, severity, and prognosis of the disease. And it should have a high sensitivity and specificity, and should be thoroughly validated before it's used in clinic, and also cost effective. Type of biomarkers, actually there is a lot of biomarkers, but the most important are the biochemical markers, the molecular markers, and cellular markers, and others markers like the microparticles. So in this talk, I will specifically talk about the microparticles, and my colleague, Dr. Despina Sanudu, will have prepared a great talk on microRNA, so we'll hear from her. The biochemical markers, in the last year, I have highlighted the importance and the value of the biochemical markers in heart failure, so I'll be focusing more on microparticles this time. So what are the microparticles? They are small vesicles shredded from cell membrane in response to numerous conditions, mainly from activation or apoptosis. Microparticles are released from different cell types in circulation, mainly from platelets, endothelial, leukocyte, red blood cells, and others. During their formation, they retain membrane receptors inherited from their parental cells, as well as part of the cytoplasmic content. It's well recognized that formation of microparticle occur mainly by cell activation or apoptosis. In cell activation, rise in intracellular calcium induce externalization of phosphatidylserine and activation of calpain, which is necessary for cytoskeleton disruption, membrane bleeding, and formation of microparticle. In the apoptosis induction, cleavage of caspases will lead to ROC activation, and finally leads to cytoskeleton disruption and subsequently formation of microparticle as well. Numerous studies indicated that the circulating level of or the levels of circulating microparticles are increased in cardiovascular diseases, including acute coronary syndrome and heart failure. For example, the analysis of levels of circulating endothelial microparticles in patients with heart failure, we can find a huge, significant increase between the level of endothelial microparticle in a patient with heart failure symptoms compared to healthy subjects, and this increase is proportional to the severity of the disease. And when we look to the analysis of the frequency of total cardiovascular events, we find that patients with high level of EMP have a high risk of developing cardiovascular event than the group of patients with low level of microparticles. And another study actually, when we look at the event-free survival in a multi-marker score category or in the Seattle heart failure model, we found that a patient with moderate to high tertile have also more chance to develop cardiovascular event than a patient with low tertile. So here also I would highlight the importance of a multi-marker strategy in the risk stratification of patients with heart failure. So what we are doing in QCRC in terms of research on biomarkers. So QCRC has two laboratories at Qatar Science Technology Park as well as here in the Heart Hospital. And these laboratories are equipped with state-of-art equipment and technology to look specifically into biomarkers, so all type of biomarkers, using different methods like transcriptomic, proteomic, genomic, metabolomic, and as well as bioinformatic. So we are actually working closely with our colleague here in the Heart Hospital in studies about different aspects of heart failure, and specifically we are studying the role of biomarkers, microparticles, microRNA, and biochemical markers in post-primary PCI adverse remodeling in the Qatari patient population. And we aim, actually, our specific aim is to determine the predictors, the incidence, and clinical characteristics of patients who develop adverse remodeling after myocardial infarction, and to define the changes in the global biomarker expression, and to correlate these changes with clinical data, and to evaluate the potential value of biomarkers as predictors and prognostic marker of disease progression in adverse remodeling after myocardial infarction. So what is our methodology to detect those macroparticles in the lab? So first we collect the blood from patient, for STEMI patient who undergo PCI, primary PCI at different time point, and this blood is subjected to a series of differential centrifugation, and finally obtain different parts of the blood, and we are looking to the microparticle and the other biomarker in the PPP, the platelet poor plasma, and plasma sample are then isolated and analyzed or stored for further analysis. I said before in the beginning of the talk that the microparticle inherits some proteins and specifically some surface markers from the parental cell. And these surface markers will help us to identify specifically a specific type of microparticle. For example, for platelets express a panel of surface markers that are exclusively expressed in platelets, and we can use some monoclonal antibodies that is specifically directed against these markers. For example, CD41 and CD61 as well as CD42B are specific markers for platelets. Same for the endothelial cell and different population of leukocytes. So this will help us to actually identify specific subtype of biomarkers. This is typically what we get in flow cytometry. This is a typical plot of flow cytometry, we can draw a gate for microparticle using a calibrated fluorescent beads, and this will help us to draw a region, and we expect having the microparticle into this region. And this is what we got from the plasma sample without staining. And for example, here, if you want to detect the platelet microparticle in particular, we can use CD41 antibodies, and then we can make the difference between platelets and other population of microparticle. And here also, we can identify in one run in the flow cytometer two different populations, endothelial microparticle and platelet microparticle. So the anticipated result, we seek to demonstrate that myocardial infarction is associated with specific changes in the expression of circulating biomarkers, and these changes will be correlated with the clinical data obtained to determine a panel of markers that serve as prognostic indices for adverse remodeling after myocardial infarction. And we believe that this will have a significant impact on the determination of the incidence, severity, and outcomes of myocardial infarction in the Qatari population. And also we believe that microparticles play an important role in diagnosis and prognosis in heart failure, and in conjunction with other biomarkers, MPs and detection of microparticles may open the field to the development of strategies for biomarker-guided therapy. Thank you very much for your attention.